All right. Happy Friday. End of the week. Next week's halfway. Next Wednesday's the halfway mark. How did lab on Wednesday and Thursday go? How many grasshoppers you get? All right, so yeah, that lab's posted. The Excel sheet's posted. The data, we'll post the data in the lab itself. All right, so we'll all have the same data. We'll calculate population estimates. And then what we'll do is we do a simulation in the lab where we assess what happens when we violate assumptions of that Lincoln index. So usually the lab takes most of the time because the simulation, in order to get better estimates and have a better idea of what happens to our estimate when we violate assumptions, we pool the class data. So each group, each pair does their own simulation. We collect the class data, and then we calculate the average population estimate. And that means then that when we get out, depends on the slowest group. So come ready to work. The quiz. So I posted the quiz on the aquatic systems. I think I put that due end of the day today. Kind of keep up with it because we'll get through most, if not all, of this presentation today, this intraspecific variation. We may go into Monday, but then that quiz goes live on Monday, and then measuring variation goes live. Will probably be due by the end of Friday. And that gives us time then to get ready for demographics. So, any questions? All right, we're going to start here. Sources and causes of variation. So we've talked about evolution side of things. Where we're moving is talking about, what environmental factors could produce our variation that we see across the species, and even maybe between populations, between species, and so forth. So, just as a review, you know that the environmental variation that's out there is caused by the discontinuities in both the physical and biological factors and resources. We talked about this for the terrestrial systems. We talked about it for the aquatic systems. And this is really just saying that, hey, what the environment looks like over here is different than what it looks like over there, and it's because of variation in the landscape, variation in weather patterns, and so forth. All right, so because we have this variation across our landscape, this is the geographical abiotic variation, we then see the variation in our populations and variation in our communities. Now, the environmental variation tends to exist as series or perhaps collections of habitat patches. So you have to think like on the large scale, broad scale, to recognize that suitable habitats occur in isolated areas. All right, and we call those habitat patches. And each of those patches could have its own set of abiotic and biological factors. The question then becomes, how do those populations in those patches respond? All right, well, what we see is that we see populations different. They vary. They vary in their phenotypes. And the phenotypes isn't just what we can see. So their morphology. It could be variation in their behaviors, in their life history strategies. Why does that variation exist? Well, it exists because each of those patches has its own set of selective factors, selective forces. And evolution then has shaped and produced those phenotypes that are going to be best adapted for our environment, or at least, they might not be best overall, but it's good enough to where they outcompete all other patches. So what is this intraspecific variation that we're talking about? Well, it has a specific term. It's that genetic and phenotypic variation within a species. So we're talking about within a species, Why do we see the variation between the populations? All right, so intraspecific variation is the variation within a species, and this is both genetic and phenotypic variation. We're going to talk about the factors that influence this variation. All right, so we are influencing the variation through spatial and temporal factors. All right, at the spatial level, that variation that we see across the species is due in part to the magnitude of differences between our patches. So you can say, how different are they? Are we going from a hardwood forest versus a pine forest? Yeah, that's kind of a difference. Are we going in a habitat patch that receives 100 centimeters of rain a year versus 20 centimeters of rain a year? That's a pretty big difference in rainfall. All right, so magnitude difference between the patches. The degree of isolation between the patches. Now, hopefully you kind of recall what we talked about gene flow and some of those ideas related to gene flow and like shifting balance theory and all that stuff. We're coming back to that. And then also relative size of the patches. How big is that habitat patch? Is it going to support a large population size, or is it supporting a small population size? Why does that matter? Again, think back to genetic drift and what we were talking about, the effect of genetic drift on our populations. In terms of temporal variation, we're talking about the pattern of variation. Is it predictable or not? Is the pattern slow ramping up? Is it very quickly and happens over a very short period of time? Is the pattern going to be a regular type of pattern, so continuous increase and decrease? You can think daily temperatures, warms up during the day, cools off at night. All right, that's our pattern. Or are we talking about something where it is always going to increase and then maybe stop for a long period of time, and then we have this decrease? So there's a lot of things to consider when trying to explain the intraspecific variation across a species. And what we're going to do is we're going to touch on each of these. All right, you ready? All right, so don't start your quiz yet, but our answer, at least one of the answers for our quiz, is banana. Answers are all fruit. Banana. Write that down. Banana. We'll later on we'll take a break and take the quiz, and you'll get the other one, if there is another one, or two, or three. All right. Spatial variability. We're going to start with the magnitude of differences between the patches. This is related to our selection pressures. So the more different they are, the more different selection pressures. So then in theory, what we see, the more different the patches, the more different those populations are from each other. That boundary layer between our patches, that transition zone, has a name, and it's called the ecotone. So the ecotone is the boundary between the various habitat types. This could be a very gradual change, or it could be a very abrupt change. So here in this image, we can see what I would call two different habitat patches. All right, you've got more of a soil type environment, and then you get to a rock, large rocky surface. That transition zone between the soil and the rock, that transition is the ecotone. And that is a very abrupt change. All right, contrast that with some of the sandy shorelines. the dune type areas. You're going to go from sand to rich soil, all right? And in a natural setting, that's a gradual change. You'll see 100% sand, you'll see over here 100% soil, and there's a zone where you've got both sand and rich soil. Because that rich soil is kind of developed and built up because of vegetation that has grown and died and is leaving organic matter. So you've got this gradual change in that type of environment, whereas this environment is much more abrupt. You've got soil and then you have rock. Okay? Now I don't want you to think about plants living on here. Consider an insect, or maybe a lizard that forages. That lizard could be foraging in this type of soil environment, or the lizard may forage on this type of rock environment. We have a very abrupt change between those two. So if the lizard populations, if one lizard's population is down here and another is up here, how do you think that ecotone, that narrow, that abrupt change, affects our variation between those lizards? Which one is going to lead to more variation? That very abrupt change or the gradual change? What do you think? As this, again, intraspecific variation, so differences between our population. We got our two populations. Which type of boundary will produce more variation? Why abrupt? Yep. So that's the idea. So the abrupt change is going to lead to more intraspecific variation because you don't have any of those in between. You're either adapted for this habitat, or you're adapted to this habitat. There's no intermediate. If we go to the sand dune type area, you're in the sand, or you can be in the good, rich soil environment, which you can think of as more vegetation. All right, you have those populations at the extreme, but some of those variations, some of those adaptations that those two extremes have, will also work fairly well in that gradual transition zone. And if they can work well in that gradual transition zone, then you can start to have some sort of interbreeding between those populations, or even migration between them. So that movement then can help neutralize some of the selective forces and reduce the amount of variation that we see across that species. So the ecotones, that boundary, that change can be very abrupt or it could be gradual. Second part is isolation. Degree of isolation of our patches. So some of our patches are close together, and that was this example. They're close together, or our sand dune type area could be close together, or they could be far apart, say at opposite ends of the state. How does the degree of isolation affect our intraspecific variation? Which situation would produce more variation if we're close together, if our patches are close together, or if they're isolated? Did you say something? Whisper. Share the wealth. Turn to your neighbor. Pick an answer. Isolated or close together? Colby, you're like isolated back there. Should make you move. All right. What do you guys come up with? Close together would produce more variation? Okay. Does anyone have a different answer? That's a good one. Okay, so we've got this idea that if you're close together, you can have more interaction between the populations, whereas if you're isolated, you have less interaction. What's the interaction that we could have? Breeding. Breeding. What process of evolution is that? We talked about 4. Spent more time on one of them. Gene flow. Our interaction that we're talking about is gene flow. Gene flow neutralizes our variation. So if you're close together, you're more likely to have gene flow between those populations, which now will start to spread the genes between the populations, and in many cases homogenize the variation across that species. If you're isolated, the chance for gene flow is much, much less, and thus you can kind of selection can work faster. Remember when we talked about gene flow can oppose or reinforce selection? This is what's going on. If you're close together, gene flows could be opposing it because we're neutralizing that variation. We're breaking up the good gene combinations. Whereas if we're isolated, yeah, selection's going to be selection's going to be working quite effectively. Should be quite effectively. All right, so degree of isolation. I have the Galapagos Islands here because it's a high spot at biodiversity. Why does this area have a lot of biodiversity? Just talked about it. Because the ocean currents, right? Ocean currents, this is a site of upwelling. A lot of primary production is going to be occurring there, which now is going to support a large group of organisms. But then the other aspect of it, because you have to think, well, it's also happening at the coast of South America, right? South and Central America. You have those hot spots. But why here? They're isolated. They're isolated. So selection's going down. You don't have gene flow with the mainland and stuff. So you can have, more speciation events happen out here because we are isolated. Sizes of the patch plays a slightly different role. So we're going to talk about size in terms of population size first. All right? And this one, we'll come back to it when we talk about measuring variation. So if you have a large patch, you can support a large population. If you have a small patch, you can support a small population. All right? Drift is more likely to occur in those small populations, or at least the effective drift is going to be more likely. So if you have habitat patches filled with if you have groups of small habitat patches supporting our species, you could produce more variation just because of drift. All right? But that's only one component. The other component is related to the mobility of the organism. That's what this slide is presenting. All right? It's what does the organism see? What does the organism experience? Does it experience a habitat with a lot of different, what you could say, microhabitats? Or does the organism, wherever it's at, only experience one type of microhabitat? The terminology for this is called grain. What is the grain in our environment? Now this goes back to the idea of photography and film. Because those light particles that developed film had different grain sizes. And this is probably, I guess if anyone has taken like art, maybe you've covered it. Maybe you covered this. So to change from the old film type example to TV type examples, consider grain the difference between a 260p on YouTube versus your ultra high definition YouTube feed. All right. Coarse grain variation is spatial variation that is large relative to the mobility of the organism. All right. This is, if you can think about it in terms of, streaming services, this is pixelation in your video. You can see the pixels. All right. You can see the pixels, and our organism is going to be basically in one pixel. That's it. So that organism is going to only experience one or maybe a few environments over its time. Right? And when you see it, it's, videos aren't very clear. All right? It's just pixel. It's just pixel. Fine-grained variation is when our spatial variation is small relative to the mobility of the organism. Now, the individual pixels on our ultra-high definition YouTube stream is super small. So you can see extreme detail. Extreme detail in that video. And when I tell you that with the 760 high definition TVs came out, and they marketed it as you can see every blade of grass on the TV. It's like, yeah, that's great if you have the actual broadcast in high definition. All right, but you've got coarse grain is very broad, and our organism then only sees one or a few habitat types in their lifetime, and the fine is very, very clear. And in this case, then, our organism is going to see many different habitat patches, not only through its lifetime, but probably on a daily basis. All right, so this is kind of the image here. We plotted, let's say, a fairly large area of land. Let's go out to, a 50-acre ranch. We might be able to mark off that ranch to have 5 different habitat patches. All right, and that's what the cross-hatching and the non-shading. Depending on the organism, our organism may only be restricted or may only see one type of habitat patch. So consider this. Maybe this is a land snail or a small mouse. They only see this. They see their environment as being very coarse. To them, the environment is coarse grain variation. Versus a hawk that flies overhead looking for that mouse, they see that environment as very high detail. It sees all of these variations, and it experiences all of these habitat types. Now, the question then becomes, which type of variation leads to more intraspecific variation in our species? The coarse grain variation or the fine grain variation? Well, it kind of harkens back to these last 2 points. Because if you only experience one habitat type in your lifetime, then selection is picking the phenotype that is best suited for that habitat type. So if you have individuals living in this habitat patch, they're going to get a phenotype that's adapted to that environment. Populations in this habitat type, they develop adaptations for that environment. So you can end up getting then multiple populations that are adapted to their own special environment. Do you think that's a lot of variation, a lot of intraspecific variation? Yeah, it is. Should be. We should see a lot more variation in coarse-grained environments. Versus a hawk, where today he's got these selection pressures, tomorrow he's maybe down here. Right? The selection pressures aren't favoring any one specific phenotype that's going to be best for one environment, because he might not be there tomorrow. He could be in a different habitat. So, in terms of evolution and how it works, it's looking for a balance of phenotypes. It's looking for a phenotype that can at least do well in all of these habitat types. Are we going to see a lot of variation across the species? Probably not. Probably not, because our species is experiencing a lot of habitats, a lot of environmental variation, and evolution then will favor those phenotypes that at least do okay across the board. All right? So make sure you have an answer to that in your notes. Questions? Temporal variation. So our environmental variation can vary in time. So far, all of these, we've just talked about everything stationary. It stays the same. But we do know that it's going to vary in time. It can vary in time. And we talk about pattern, speed, and predictability. Those are the main ones. So the pattern could be daily or seasonal. And it could be a large or a small change. In terms of speed, it could be fast or it could be gradual. And this variation could be predictable, could be a predictable change, or could be unpredictable. So let's talk about how each of these affects our intraspecific variation. We should probably make a note when dealing with temporal variability. Our answers will rely on generation times. And generation time, we'll have a definition for it. Generation time is basically how long it takes, the age of when our new cohort starts reproducing. We'll get to that in the demographics. We actually have a lab that works through life tables. But in terms of generation time, we're talking about how long does the organism live? All right, because a long-lived organism, let's say humans, all right, we are going to experience a lot of temporal change. Daily changes, seasonal changes doesn't really do much for us because we live so long. You know, anything that we see this winter, the selective factors probably are very minimal. All right, instead, we're looking at these changes over, let's say, 20-year time spans, 30-year time spans. All right, and that's different from a bacteria or a yeast that may have 20-minute, maybe 2-hour generation time. All right, so you have to kind of keep those in the back of your mind, and that's kind of the daily versus seasonal. Let's talk about magnitude of change. All right, if we're going to get a big change versus a small change, which one do you think is going to produce more intraspecific variation? Big changes or small changes? Big changes, why? Significant enough. Yeah, so big changes mean bigger changes in our selection pressures. Basically it. I mean, what you said is 100% correct, but I want to put it in terms of what we've been covering, what we've covered. So a large magnitude change, going from average daily high of 80 degrees to an average daily high of 50 degrees, that's a big change. That's going to produce a very different selection pressures, which is going to increase our variation. Compared to a daily high of 80 degrees versus 79 degrees, not much of a change at all. Speed of the change, it could be fast or it could be gradual. Which one would produce more variation? What's that? Fast? What fast? Yeah, I understand where you're going, and I'm gonna change it a little bit because I think your explanation's kind of similar to the ecotone. So abrupt versus gradual change of the ecotones. I'm gonna change it in terms of evolution and inheritance. So gradual change can lead to more easily adaptation because our adaptations tend to be gradual in time. Fast changes, an organism might not be able to adapt, and that could wipe them out. That could wipe them out. What about predictability? How might that? And we'll be coming back to these too, because we need to. Where we're going is if two populations are different, genetically different, what could produce that? And we're coming back to all these ideas. So which one would be easier to adapt to? And that's the same idea with the fast versus slow. Which one's easier to adjust to? Which one? Yeah, the predictable one. I already have it here. Unpredictable changes, you can't prep for it. You don't know it's coming. You don't know it's coming. So if you have an unpredictable change, it could wipe you out, never give you a chance to evolve. All right. So now let's talk about the pattern of variation. Let's talk about our individual responses. So recall species. That's a group of populations that can interbreed and produce fertile offspring. I think we may have said that. Now we have a formal definition. Definition is now fair game for our exams. All right, subspecies is a local phenotypically distinct form. Some people have called it a race. All right, usually these subspecies exist in different geographical areas. And I could ask you, why do they occur in different geographical areas? And hopefully you could say it's because of environmental variation. Our subspecies have been adapted to the variation that they encounter, that they see. And that's both spatial and temporal variation. So this is a great example. These are snails. These are all the same species of snails, yet they have different color variations. Right? What we see is that the differences in our selective pressures produce this. And the selective pressure for this snail species is the predator. So across our geographical landscape, where these occur, you have different predators. And you have different predation risks. So the selective forces try to get these guys to hide from their predators. So whatever the dominant vegetation, whatever the dominant background is, wherever that population is, that's what selection is producing. So you have the shell that tends to color match whatever that background vegetation is, because if you matched it, it's harder for the predator to see you. You thus have higher fitness. All right? So when we look across this phenotypic variation across the landscape, it tends to correlate with the environmental variation. It tends to correlate quite well. Now this variation could arise in one of two ways. It could be that nurture part, so where you grew up could affect your phenotype, or it could be nature, and this could be entirely genetic. So in our talk, if we are nature, we're talking about the formation of ecotypes, which are genetically distinct, right? Genetically distinct populations. If we're talking about nurture, then this variation we see is entirely due to phenotypic plasticity. In 2 R's, can I freeze this. Just leave that up there, freeze, so you can see it. Actually, the people at home can't see it. I missed that. All right. Better. All right, you ready? All right, ecotypes. We've defined those, right? So you can go back to exam 1 material, look up that, get that definition, get it moved over as well. Our example of ecotypes are in serpentine soils. So what are serpentine soils? These are soils that are high in magnesium. They're going to be low in nutrients, so nitrogen, phosphorus, potassium, and they have a poor water holding capability. Right? Not a very good soil for plants to grow in. When we see these areas, right, the serpentine soils exist as patches across the landscape. So it's not like one massive area of serpentine soils. You see the serpentine soils in patches. Some of the plants grow in those serpentine patches. Others, they grow outside of those patches. And we're talking about right here. In this floor, we've got the tile, these 1 foot square tiles. If this is our entire landscape, we've got different tiles, or maybe groups of tiles with serpentine soils. Other groups, not. They've got great or good soil. Yet we have the plant species that when we look at it, we've got some that are growing in serpentine soils, some that are outside. When we look at these plants, you could say, well, this is just the same. This is the same species. And it is. But then you'd also say, given their location, they're so close together, surely they're breeding together, right, and producing offspring, so genetically they should all be similar, but you would be wrong. What we see is that we have distinct ecotypes in this landscape. Those plants that can grow and survive in the serpentine soils are genetically distinct than those plants that grow and survive in the non-serpentine soils. Why would that be the case? Why would they be genetically distinct? Why are they distinct? Okay, what do they have to deal with? Yep, and actually, magnesium's the main driver. So these ecotypes, it's not just, you have the selective force. When we actually look at how these plants have adapted, they have adapted a way to utilize magnesium. So magnesium is 2+, cation. Those are, it's right up there. 2+. All right. What other 2+ molecules often used in metabolic processes? What were you going to say? Calcium. Good, good. I thought you were going to say like beryllium. Calcium, right? So these guys have found ways. They've evolved ways to utilize magnesium in place of the calcium to kind of get around that toxic component. Some of them, they take the calcium in and try to sequester it to minimize its actual impact. That requires a lot of metabolic pathways. They had to evolve, and thus you needed the genetic code to do that. There's our ecotypes. Now, what governs the evolution of these ecotypes? We've got organismal factors and we have environmental factors. All right, so at the basic level, in order for the ecotypes to form, heritable variation must exist. And that goes back to Darwin's tenets. In order to produce genetic differentiation, we need that heritable variation. That variation that is going to be passed down to our offspring, to our grand offspring, and so forth. Right? Fisher actually developed a theorem for it. This is Fisher's theorem. Fisher's fundamental theorem. Fundamental Fisher's theorem is how you can also see it. So the rate at which the mean fitness of the population increases by natural selection is equal to the additive genetic variation in the fitness. We discussed this without using this definition. The speed, the rate of change in our population is determined by the heritability. If you have a high heritability value, you can see a faster rate of change in our population. What was heritability? It measured the amount of additive genetic variation. And what was additive genetic variation? It's the combined effects of all of the different genes that affect fitness that are or can be inherited. So if selection is going to produce these ecotypes, we have to have that heritable variation that must exist. And the more the phenotype is determined by the additive effects of the genes, the faster we can produce these ecotypes. And the easier it is to produce these ecotypes. In terms of the environmental factors, we've got two things: the intensity of selection and the barriers to gene flow. So again, we talked about rate of change. We said it depended on heritability, that was H. It depended on the selection coefficient. We've already talked about this and said, if you increase the selection coefficient, the selection pressures, you will get a faster rate of change. So if we have two habitats and they differ so much that the selection pressure is great, like our serpentine soils, magnesium's toxic. It's going to be at high selection pressures. You're going to see a stronger push to produce these ecotypes, or to produce a ecotype. Likewise, we can't have selection working and pushing us one way only to be countered by lower fitness alleles coming into our population because of gene flow. All right, so if we're going to go for ecotype formation, one, we need heritable variation. We need selection. Stronger selection there is, the stronger we're selecting for an ecotype. And we can't have gene flow busting all of those good genes up. Questions? Quiz. You can pull it out and start answering. There's only one answer for that quiz. I was going to ask, choose the one that is different from the rest. But I know some of you like bananas. I don't like them at all. The runts, yeah. I just trash all of them. Actually, my kids eat those. Least favorite fruit. So, answer was given. Jeez, thanks, Lily. Yeah, it was already said online. Banana. It's your answer. So, all right, did you get it? Something in the news? Pretty big? World Health Organization recommends a groundbreaking malaria vaccine for children at risk. Pretty groundbreaking. You don't have vaccines for parasites. Parasites are actually really good at what they do. Right? They've had to deal with the immune system for thousands, millions of years. Now, what I haven't seen yet is this part. For children. Because I'm sure there's going to be some people on social media. Why did the children get it? Why don't the adults get it? So we have, who's in parasitology? Oh, just one. Right? Oh, two. Oh, yeah. Lexi. Two. Why children? Why are they focused on children first? Two of you. Phone a friend. Yeah, right in front of you. Yeah. Good guess. The children weren't exposed to it. Good guess. No, that's not. What's the #1 killer of children in Africa? Malaria. Malaria kills a lot of people. Vast majority of them are kids. So, and also, don't we develop acquired immunity in malaria? We do. So repeated exposure, if they can survive it, all right, they can develop immunity to where they're not actually immune. You've had the malaria parasite circulating, but you no longer show those symptoms. And then if you leave for, let's say, a year or two and go to, let's say, schooling, come back to that area, you can get hit again. So again, that's just kind of when you say, well, shouldn't we have had antibodies against this? Yeah, parasites are really good. So we'll see how this does. They've been trying to develop for years. This is the first one. So you've got 4 doses in children. They're targeting 5 months of age all the way up. Newborns, if they are breastfed, do have some protection coming from the mother. So pretty cool. Pretty cool. Not necessarily ecology, but you know what? Hey, it's biology. Parasites. Parasites are cool. All right. All right. We'll stop here. We'll talk about phenotypic plasticity. Yeah. So we didn't quite get through all of this. So Monday, this quiz will go live, and then we'll talk about measuring variation. How do we distinguish between the nature and the nurture components? All right.